Very good afternoon everyone. Today we are going to discuss about genetic disorders. So genetic disorders are the diseases which caused by a change or abnormalities in gene or chromosome. So obviously there is a change in chromosome or gene function. Bas, that's it. So genetic disorders are usually classified into Mendelian disorders and chromosomal disorders. So we will talk about these chromosomal disorders in the next class. But today we will discuss about these Mendelian disorders. Mendelian disorders. So the name itself says that it follows Mendelian laws. And these are single gene mutations, which means there is a mutation or a change in a single gene. I will tell you an example, a general example. So you went outside with your father. So your mother wanted to text you, bring some milk from the shop. So instead of M in the milk, she texted you yes. So read the sentence. Bring some silk from the shop. Just one word, just one letter. It changed the entire meaning of the sentence. In the same way, a change in single gene will lead to change in function of the entire gene, and it leads to Mendelian disorders. So Mendelian disorders are divided into autosomal disorders and sex-linked disorders. So how many pairs of chromosomes do we have? Yes, 23. In that 23, how many of them are autosomes? 22. If there is any defect in those 22, it leads to autosomal disorders. If there is any defect in the remaining one pair, it might be XX or XY, it leads to sex-linked disorders. So again, it is classified into dominant and recessive. Dominant. So how many alleles do we have for a single gene? So normally we get one allele from mother and one allele from our father. So for dominant, the mutant allele should be present. Even if one mutant allele is present in the two alleles, it can express. Those are known as dominant. For recessive, both the alleles should be present. Only then it can show its effect. And in sex-linked, we have X-linked disorders and Y-linked. Same again in this we can see dominant and recessive. And here we can't see any dominant or recessive because it is present only in males. So let's start with the autosomal disorders. We have few examples in our textbook, so we'll discuss one by one in detail. So let's start with sickle cell anemia. Sickle cell anemia. So what's the shape of our red blood cells? Normally RBC are in biconcave shape. Biconcave. But in this disease, they will become sickle shaped, crescent shaped, means like a moon. So these are autosomal recessive, autosomal recessive disorders, which means the defect is in the first 22 pairs. And recessive, in order to express, both the alleles should be present. Both the mutant alleles should be present. Only then it can express. And the mutant allele is present in 11th chromosome. hemoglobin beta chain. So before going into this, I want to just give a general view of hemoglobin. We know about hemoglobin. It has two chains, alpha chain and beta chain. Hemoglobin. So it has two chains, alpha chain and beta chain. So this alpha chain is controlled by HBAG and this beta chain is controlled by HBBG. And this gene is present in 16th chromosome. And this gene is present in 11th chromosome. So there is a defect in this gene. Alright. And then there is a mutation in this gene and it leads to sickle cell anemia. Normally red blood cells are biconcave in shape. Because of this mutation, the red blood cells become sickle crescent shape, elongated shape. So the defect is at the 11th chromosome. So coming to the alleles which are present in this, HPA, which is a normal one, and HPS, which is a mutant one. So three possible combinations can be seen. So first one, HPA, HPA. What will it become? Normal. HPA and HPS, what will it become? It will become carrier. And HPS and HPS, so it will become deceased. So these are genotypes and phenotypes of it. HPA, HPA, if two alleles are HPA and HPA, it is a normal one. If one allele is HPA and another is a mutant one, it will become carrier. And if both the alleles are mutants, then it will become the deceased. So this is the reason why this is a recessive dysfunction. And if we draw, let's draw Punnett square for this. For carriers. For carriers, let's draw Punnett square. So parents are carriers. HB, HBS, HBA, HBS. So male, female, HB, A and HB, yes. Here also HB, A and HB, yes. So this will become HBA and HBA, which is a normal one. Here it will become HBA and HBS, which will be a carrier. Here HBA and HBS, which will also become a carrier. And here HBS and HBS, it will become deceased one. So let's write the ratio. 1:2:1. So 1 normal, 2 carriers, and the remaining is deceased. So the 50% of them are carriers, and 25% of them are normal, and 25% of them are deceased. So when we take parents as carriers, we'll get 25% of them as normal, and 50% of them as carriers, and 25% of them as deceased. Let's see what happens in a gene. in normal condition and diseased condition. Sickle cell anemia. So here we are talking about the 11th chromosome, beta chain. Beta chain, in the 6th position, it has glutamic acid. Glutamic acid. So GAG is codon for glutamic acid. So when this is present, normally we will get a normal hemoglobin. And the RBC are also good shape, biconcave. So here what happens is, I told you there is a mutation in the 11th chromosome, HPV gene, beta chain. So there is a mutation. So instead of GAG, it is replaced by GUG. Just a single letter. But that's it. It becomes, this codes for valine. And this leads to long polymers. And during when the oxygen is not sufficient, it will become long chains which are sticky in nature. The RBC are biconcave shape. They are flexible. So blood vessels are very narrow. So if the red blood cells are in biconcave shape, they can easily go into it and they are flexible. But here, they are forming long polymer chains. It becomes elongated. So can the red blood cells go like if they are long? Can they enter into the blood vessels? It's not possible. So slowly. The blood vessels will get blocked and it causes anemia. And in severe causes, it causes even death. So normally in beta chain, in the 6th position, the glutamic acid is present. But here it is replaced, GAG is replaced by GUG. And this codes for valine. And valine forms long polymers and long chains, and which makes the RBC into long elongated structure which cannot pass through the blood vessels, and because of it, it leads to anemic condition. Now let's talk about another disease called thalassemia. Thalassemia. So this is also autosomal recessive disorder. So the defect is present in autosomes and recessive. So it needs two alleles to express this disorder. So just now in sickle cell anemia we have discussed about hemoglobin. So there we have alpha and beta. Alpha chain which is controlled by HBAG, which is present in the chromosome number 16. Just now I told you about this. And here beta chain is controlled by HBB gene, which is present at 11th chromosome. So now the defect is in this chromosome and is in this chromosome. So it leads to less production of alpha chain, and there is a defect in this and it leads to less production of beta chain. So when there is a less production of alpha chain, it becomes alpha thalassemia. And when there is a defect and when there is less beta chain is present, it becomes beta thalassemia. So thalassemia, which is an autosomal recessive disorder, means the defect is present in the autosomes, and it is a recessive disorder, which means it needs both alleles to express. So hemoglobin has alpha chain and beta chain. So alpha chain is controlled by HBA gene, which is present in the chromosome number 16. So when there is a defect in this gene, mutation in this gene, it leads to less production of alpha chain, and it leads to alpha thalassemia. So the beta chain is controlled by HBB gene, which is present in the 11th chromosome, and here the beta chain production is reduced and it forms beta thalassemia. So what's the difference between sickle cell anemia and thalassemia? So sickle cell anemia is a qualitative disease, which means there is a difference in quality. There is a problem with quality. Here the problem is with quantity, less production. There the abnormal hemoglobin is formed, so it becomes quality. Here the less production of hemoglobin is formed, and this leads to, this is known as qualitative and it is quantitative. So when there is a less production of alpha and beta chain, it leads to less production of hemoglobin. So what happens if the hemoglobin is less? So what's the main function of hemoglobin? They usually transport oxygen from lungs to all parts of tissues, right? If the hemoglobin is less, the oxygen transportation will be reduced. So it causes anemia and severe other diseases. So let's draw the Punnett square for this. Let's take the parents as carriers. HB, HBD with HBA, HBD. HBA and HBD. HBA and HBD. It will become HBA normal, and this will become carrier, and even this will become carrier, and this will become diseased. So same, 1:2, 2:1. The 1 is normal, and the 2 is carriers, this 1 is diseased. So the disease can be occurred only when the parents are carriers. This happens the same thing in sickle cell anemia. So now let's talk about another disease called phenylketonuria, also known as PKU. hydroxylase. And this tyrosine produces many important substances. Like melanin and many other neurotransmitters. So in this condition there is a mutation in the 12th chromosome. So in this disease PKU, phenylalanine. So here this enzyme phenylalanine hydroxylase, which converts phenylalanine into tyrosine, is absent. So the phenylalanine cannot be converted into tyrosine. So now what will happen? Accumulation of phenylalanine will happen. Accumulation. And this accumulation leads to formation of phenylalanine acid. And this acid is produced along with few substances which are harmful in nature. And this leads to mental retardation, and formation of urine with acid, phenylalanine ketogenic acid. So phenylketonuria, also known as PKU, here the defect is present in the 12th chromosome, which is an autosomal recessive disorder. Normally in our body we have phenylalanine. So it is converted into tyrosine with the help of phenylalanine hydroxylase. So this tyrosine is important substance which produces melanin and some other important substances and also neurotransmitters. And this phenylalanine in disease condition, here the phenylalanine hydroxylase, which is an enzyme which converts phenylalanine into tyrosine, is absent. So now tyrosine is not formed. Phenylalanine will remain. So it will accumulate and it will form phenylpyruvic acid. Phenylpyruvic acid, so which is a harmful one, and this leads to mental retardation and urine with pyruvic acid. So that's it about phenylketonuria, and we will discuss about the remaining disorders in the next class or in the next video.